Yesterday, we'd gotten as far as a description of the neuron doctrine. We defined the neuron doctrine saying that the neuron is the fundamental structural and functional unit of the nervous system. And I wanted to give you a little history on that today, tell you where that idea comes from. It really is a foundational principle for all of neurobiology and has been for the last 120 years. And this foundational principle has to do with the idea that the individual neuron is the cell that is functional in the nervous system. Now, we'll see as we go through this, and particularly when we talk about glia tomorrow, that story is not quite as clear-cut as it used to be. But for the most part, we still base most of our thinking in neurobiology on this idea of the neuron doctrine. And where the neuron doctrine came from was largely from two major scientists who worked in Spain with Santiago Ramon y Cajal and Camillo Golgi, who was working in Italy. And these two guys were basically in competition with one another. And Golgi was working at the heart of scientific Europe. He's working at some of the best universities in Italy. Santiago Ramon y Cajal is working in Valencia and later in Madrid, sort of on the fringes of science in Europe. But he recognized the importance of a stain called the Golgi stain. In fact, a stain that was developed by Camillo Golgi. And what the stain would do is it would stain neurons in their entirety, so they could see the whole neuron. Up to that point, they could see cell bodies, but they couldn't see the whole neuron. And when they looked at sections through brains and looked at cell bodies, it was almost impossible to interpret. There were cell bodies everywhere. If you put your phone away, I'd appreciate it. And so then what would happen is they would see all these cell bodies, but they couldn't make much sense of the nervous system. Well, along came the Golgi stain. And this now allowed them to see neurons in a brand new way. But interestingly enough, it generated a kind of argument between Golgi himself, who invented the stain, and Santiago Ramon y Cajal, who had basically further developed the stain. And they ended up with this odd dynamic in that Golgi was a reticulist. He said, as I mentioned yesterday, all cells are connected one to another through one large syncytium. There are no differences in cells themselves. They're just all connected one to another. Whereas Santiago Ramon y Cajal suggested that each cell is a distinct unit. And because each cell is a distinct unit, he came up with an important concept, an important rule called the law of dynamic polarization, meaning information would flow in neurons in one direction, and I'll walk us through that. Now, when this was all settled in the early part of the 20th century, there was a kind of structural description of the neuron, and this largely came from Cajal's work. It said that there is a dendrite, there's a cell body, there's an axon, and there's an axon terminal. And from the law of dynamic polarization, information flowed in one direction. It flowed to the dendrite, to the cell body, to the axon terminal. And that's an anatomic description, and it works just fine in most cases. There are a few cases where it breaks down, but for the most part, it works pretty well. But then 60 years later, along came electrophysiology. And with electrophysiology, they could now begin to look at the neuron in a different light. They could now begin looking at the neuron in a way that was more functional. And so here I'll give a functional description of the neuron. And in this way, we now, rather than calling a dendrite cell body axon, we're now going to talk about an input region, a conductal region, and an output region. And one of the major insights of the electrophysiology was to determine this distribution of the ion channels in the neuron. And we'll see that as we work our way through this, that in the input region, we have predominantly ligand-gated channels that are going to be opened by a neurotransmitter. In the conductal region, we have predominantly voltage-gated channels, particularly sodium and potassium. And in the conductal region, as we work our way through this, we'll find it's these voltage-gated sodium-potassium channels that are responsible for conducting the action potentials from one spot to the next. And then finally, this is the output region. And again, this is effectively the axon terminal. But we can talk about the output region as the region where the neuron releases neurotransmitter to communicate to the next cell. And one of the upshots of the neuron doctrine is effectively these neurons are all connected in a kind of daisy chain. But the daisy chain is the connection through synapses and chemical synaptic transmission, not through electrical connectivity of the cytoplasm. And we'll talk a good bit about that. But when we draw the neuron and we look at the neuron, we find that one of its characteristics is it's asymmetric. And we've touched on this before. We talked about cytoplasmic transport of different kinds of proteins, particularly those that were processed in the ER and Golgi. We said that those proteins are transported in vesicles on microtubules. And this thinking arose from the importance of trying to sort out how proteins could be made in the neuronal cell body and delivered to all points in the neuron and in this highly asymmetric cell. The problem isn't so apparent in a spherical cell, but when you start talking about neurons which have these enormous axons or conductor regions on them, you end up with these very asymmetric cells. So that brought up this idea of exoplasmic transport. And we've delved into that a little bit, so we'll go through the first part fairly quickly. And that is, there are two kinds of transport in cells, and in these neurons, in all cells probably, and that these transport systems can be broken down into a fast transport system and a slow transport system. And what's more is the transport systems are moving different kinds of proteins. That the fast transport system is all vesicular. So those molecules that were synthesized were synthesized by rough endoplasmic reticulum, packaged in vesicles and processed with the Golgi, those vesicles are going to move by fast exoplasmic transport. Same story we talked about earlier, vesicles, kinesin, microtubules. But there's another form of transport, which is slow exoplasmic transport. And this is about 100 times slower. 0.5 to 2 millimeters a day. And the proteins now that are moving in this component are the cytoplasmic proteins. So the idea here is we have two major classes of proteins. Ones that are processed in the ER and Golgi, those guys move by fast exoplasmic transport. But those molecules that are generated on free ribosomes, those are moved by slow exoplasmic transport. And we'll see there's a similarity here, but a stark difference, and we'll walk through that. And finally, I'll talk a little bit about the experiments that were used to sort this out. There's some clever experiments that were done to distinguish fast and slow exoplasmic transport. But let's start here with the definition we had yesterday. That is the Neuron Doctrine, which, again, says that the neuron is the fundamental structural and functional unit of the nervous system. But this idea, this doctrine, was not always so clear. And if you go back to the 1890s, early parts of the 20th century, you find there was an enormous argument going on throughout biological science, trying to sort out what the nervous system was made of. The problem fundamentally was that the microscopy wasn't good enough. The problem was that in the late part of the 19th century, the early part of the 20th century, microscopes had gotten to be pretty good. The optics had been worked out, and we could see a lot with microscopes. But the problem was in looking at biological tissues. The only way you could really look at a biological tissue in a light microscope was to somehow stain the tissues. Otherwise, there wasn't enough contrast. And this gave great insights into things like muscle and into epithelia, into connective tissue. But when they started to apply these same rules to looking at the nervous tissue, when they started looking at brains, it became very confusing. Because one of the characteristics of the central nervous system is you have literally billions of cells all packed into your brain in the brain case and in the spinal cord. So there are tremendous numbers of cells. And when they started to use their classic staining techniques, and they would cut a section and look at those sections in the microscope, they would see all of these cells, and they couldn't see how they were connected. So this gave rise to two main ideas. It gave rise to the idea that all cells, all neurons, and they could see neurons. They could see neurons because they could see the cell bodies. Once school of thought said all neurons are connected to one another by cytoplasmic bridges. They basically said that the nervous system was a reticulum. Sometimes known as a syncytium. Now, this obviously had a problem, because how would you, in fact, conduct information from place to place? How would you differentially conduct information we talked about yesterday, having sensory afferents headed toward the nervous system, motor efferents away? How would you actually get directional flow in this arrangement? But that was basically overlooked, and then people from the anatomy said, all these cells are interconnected. This group of people were known as the reticularists. The other group said, no, neurons are individual cells. And they didn't know it at the time, but we'll see here probably later this week, early next week, we're going to see that these cells are going to be connected one to another at chemical synapses. They didn't know that. But it gave rise to these two schools of thought, and these guys are called the cellularists. And the argument was going on, and there was a fellow, I'll go to the upper panel, there was a fellow in Italy named Emilio Golgi. Now, Emilio Golgi was an outstanding microscopist. He was, in fact, the guy who first described the Golgi apparatus. It was barely visible at that time with the techniques they had. But he could see something in the cytoplasm, he could see it from cell to cell, and he basically described it. And from that description, it's still to this day known as the Golgi apparatus. Now, as part of his microscopy, he developed something called the Golgi Stain. And the Golgi stain was mostly chromium and silver salts. For reasons we still don't quite understand, the chromium and silver salts would precipitate in neurons. But they would only precipitate in some neurons. They would only precipitate in 1 to 2% of the neurons. Now, initially, you would think that a stain, a label, that only recognized 1 to 2% of the cells wouldn't be of much good to you. You'd only see a very small number. But it was a tremendous advantage in the central nervous system, because in the central nervous system, by staining 1 to 2% of the cells, you could now make out individual cells. And the other advantage is that this precipitation occurred throughout the cell. Where previous labeling techniques would recognize particularly the neuronal cell bodies. They were acid stains that would stain RNA and DNA, and you could see the cell bodies, but you couldn't see any connections from those cell bodies. But now suddenly this allowed for the visualization of the entire neuron. So Golgi, working in Italy, that's right. Golgi, working in Italy, used this stain, and he would stare at cells day after day after day, and he came up with the idea that the nervous system is a reticulum. Golgi was effectively a reticulus. Meanwhile, there's a guy named Santiago Ramon y Cajal. Santiago Ramon y Cajal was working in Spain. He really, as a young man, wanted to be an artist. He really was aspiring to be an artist. And his father said, no, you have to do something useful with your life. And so he forced him to go to medical school. Some things never change. And so he went off to medical school and was trained as a physician. And he then took up academic positions as an anatomist at a number of universities, first at Valencia. But Santiago Ramon y Cajal now then recognized the importance of the Golgi stain. So he modified, not much, he modified the Golgi stain. And the story is that what he would do is he would go into the lab in the morning, and he would have all of these slides that he'd prepared the day before. And these were slides that were sections of through the brain. And he would then sit and stare through the microscope, hour after hour after hour, just looking at these labeled cells. And then in the afternoon, he would go out and sit around the park, sort of hang out in the park and feed the pigeons, and then come back in the evening to the lab. And he'd come back to the lab in the evening, and he would then draw what he'd seen in the morning. So the idea here is that he was somehow using his brain as like a kind of computational device to sort out all the noise he'd seen in the morning to make a drawing of a composite cell. So he'd make these drawings of the composite cells, and these, until the late 1960s, these were the very best drawings we had of any cell. These are the ones he had done as an anatomist. And again, it was probably his background as an artist and his training as an artist that helped him really recognize how to draw these cells and make them look real. But at any rate, he spent years and years doing this. And after a long time, he said, the neuron is a unitary cell. And he said that the nervous system is made up of neurons. He fundamentally was a cellularist. In the process of exploring the shapes of these cells and describing the shapes of the cells, he came to a conclusion which turned out to be really important. It was the law of dynamic polarization. And this said that information propagates in one direction. In neurons. And he basically said that the propagation direction is the dendrite to the cell body, to the axon terminal. Cajal fundamentally set up the concepts that we still use in cellular neurobiology. Yes?
So is the law of dynamic, what's the last word?
Polarization. It's the law of dynamic polarization. And it's a bit of a misnomer at this point, because when he's talking about the law of dynamic polarization, he's simply talking about the directionality of information movement. Here in a couple of days, we'll talk about the polarization of cells in terms of ions, and the fact that we'll polarize the membrane, being positive on the outside and negative on the inside. So don't get those two confused. This was well before they had a clear indication of the electricity in cells. And in fact, they had a vague idea. Galvani and others had indicated that there was some sort of bioelectricity, but it was not at all clear how it was settled at the cellular level. That took another 60 years to sort out. So it's the law of dynamic polarization, but recognize that's a kind of an anatomic polarization and not a cellular or ionic polarization. But at any rate, he studied and studied and studied, sorted this out, convinced himself he was true, and basically went on tour. He went on tour throughout Northern Europe. And he went to Germany, and he went to England, he went to France, and he went to Italy, and he showed his data to all of these different scientists. And pretty much that was the turning point. After he went on tour, people recognized, for the most part, that the reticular hypothesis of the organization of the nervous system just wasn't right, and that the cellular organization was the correct way to think about the nervous system. Well, the story continues so that the important contributions of Golgi in terms of the stain and the important contributions of Cajal in terms of the interpretation were awarded the Nobel Prize in 1906. And if you go back and read the speeches, the Nobel Prize speeches that they gave at the time, it's really interesting because Golgi stuck to the reticulus theory. He basically got up there and gave his talk saying, I'm right, Cajal's an idiot. Cajal never forgave him. Cajal was bitter about this to his dying day because, after all, Cajal had worked at the fringes of science and fringes of the academic groups in science in Europe, and had developed these ideas, had developed these hypotheses which were correct, and then he was being dissed by Golgi. It's a pretty angry sort of interaction between the two of them. And Golgi went on. Golgi became fairly famous in Europe, both in the academic realm and also in politics. He became a politician. And because of his level of power in Italy, these wrong ideas of the reticulus theory were taught in medical schools well into the 1930s. 30 years after they were debunked every place else. But because of his powerful status, both within the academic community and within the political community, these wrong ideas persisted. I think that's sort of an interesting story on how there is politics in science, no matter how much we like to think it's not true. But let me show you a picture here, because I sort of like this picture. Now, why this was on a postcard is beyond me, but this was an old postcard. And the left-hand side, as you're looking at it, these are drawings. These are the drawings that Cajal made. And these are drawings from the cortex of a pigeon, perhaps one of the pigeons he was feeding in the afternoon. And what you're looking at are the Golgi stains of a variety of different cells. But what to look at here is there are these dendrites out here, these sort of these tree-like structures. Then there's a cell body, and then there's an axon. There are a series of dendrites in this pyramidal cell. There's the cell body, and there's an axon. To this day, there are neurons we see in the nervous system, which we'll describe as he did, such as these pyramidal cells. And this is a picture of Santiago Ramon Cajal sitting in his laboratory in front of his microscope. I imagine him looking at the floor thinking Golgi was a fool. But nonetheless, he got it right. And he really did change the history of how we think about neurobiology. And commonly, we think about Cajal as being the founder of modern neurobiology. But let's move on from that to describe a little bit about the structure of the neuron. And the structure of the neuron I'm going to describe here, which is the anatomic description, is one that was generated largely from Cajal's work. And it gives a certain description of the neuron, and we still use these terms commonly today. So we'll take this panel and we'll describe it, we'll label it as the anatomic description of the neuron. Now, I'm not going to draw a very complicated neuron. Most of my neurons look like pineapples, but it's a structure out here. Now, in the anatomic description, this is the dendrite. And the term dendrite comes from the Greek, from dendros, meaning tree. So they looked at these sort of filigreed structures at this end of the neuron. They said, it looks like a tree, it's got a bunch of branches on it, so they called it the dendrite. They recognized early on, even before Cajal, that there was a region of the neuron which contained the nucleus. This region of the neuron where there was the nucleus, they called the cell body. In some areas, you'll see the cell body referred to as the soma. So the cell body was then connected to the dendrite. And then emanating out of the cell body was an individual process, which they termed the axon. And finally, they said down at this end, there was the axon terminal. So the anatomic description of the neuron says there's a dendrite, there's a cell body, there's an axon, and there's an axon terminal. And the law of dynamic polarization holds that information, you can read signals if you like, move or propagate from the dendrite to the cell body, the cell body to the axon, to the axon terminal. So there's a unidirectional information flow. And the unidirectional information flow is in this direction. Let me put this above. And again, none of the scientists in the early part of the 20th century really understood how this information is going to be propagated. They understand the basis of the information. But because of their understanding of anatomy, they concluded the information must flow in one direction. This was a real problem for the reticulus because they had no way of describing how information would flow in one direction to another, how it would flow unidirectionally when every cell was connected to every other cell by a cytoplasmic bridge. And that was the question.
Does whatever the message is saying happen when it reaches the terminal, or is it happening the whole way down?
So the question here is about the message. Let me cover this fairly quickly because we're going to come back to it over and over again. This message out here is, so in this region, in fact, I can maybe use this to bridge to the next sequence. In this region, we now can call this the input region. And so in the input region, we're going to receive information. We say signals. We'll receive signals from neighboring cells. we're going to call, we'll come back to this, but we may as well fill it in here. We'll call this signal at this site a synaptic potential. And the synaptic potential will be caused by the inputs of maybe several hundred other cells onto this site, all by synaptic transmission. Again, we'll come back to that later. This signal, the synaptic potential, is initiated by chemical signals. These chemical signals are initiated by neurotransmitters. So all of the signaling here in the input region in the cell body, or the dendrite in the cell body, is all done by chemical signals. Then this region here is where we're going to propagate action potentials. And one of the important concepts we'll try to drive home next week is the difference between these synaptic potentials, which are initiated by chemical neurotransmitters, and action potentials, which are initiated by changes in charge distribution across the membrane. And we'll talk a lot about that. And then finally, the signal at the axon terminal is to release neurotransmitter. So that's by way of trying to explain what the signals are in these different places. But let's kind of recapitulate that on the upper panel by describing what we're going to label here as the functional description of the neuron. And in this first iteration, we're simply going to draw the neuron as we did above. So if you hold on to the idea from the above panel that we have an anatomic description of dendrite cell body axon, that's all you really need. But now let's refine that and include some of the terms we have above. So let's describe here the functional description of the neuron. I'm going to make the same drawing. We'll find that there are some exceptions to this, but we can, for purposes here today, we can make the same drawing. There's the nucleus. Now, in this functional description, as I implied in the upper panel, this region described by the dendrites in the cell body now is going to have a new name. This is going to be called the input region. The input region obtains its name. It's named in this way because all over the cell body and these dendrites, we're going to find chemical synapses. Because we have these chemical synapses on the input region, we can describe the input region as containing something known as ligand-gated channels. In other words, these are the channels that are opened by the neurotransmitter. So again, to answer the question that came earlier, in this region we have a signal which is generated by the opening of channels by a neurotransmitter that has been released from another cell. Then in this region here, the region that corresponds to the axon in the upper panel is said to be the conductile region. And the conductile region propagates it propagates all or nothing signals. Called action potentials. So again, the synaptic potentials which result from electrical ions, or which result from the movement of ions across the membrane due to the opening of ligand-gated channels. In other words, synaptic potentials are generated by a neurotransmitter. Those synaptic potentials are confined to the input region. Then we have this great long extension of the cell, which we described as the axon above. We're now going to rename this a little bit, called the conductal region. The conductal region now is propagating action potentials. And these action potentials require a specialized set of channels called voltage-gated channels, where these channels are opened by neurotransmitters, chemicals, if you will. These channels are opened by a change in charge distribution. Across the membrane. And finally, here, this is now going to be called the output region. And the output region is the site from which neurotransmitter is released. We'll find out a little later that one of the hallmarks of this region will be so-called voltage-gated. Voltage-gated calcium channels. So what I've tried to do here with these two figures is draw together the question that was asked earlier. What's the difference between the simple anatomic description and what kind of electrical potentials are propagating in those anatomical regions? So again, the signals here in the input region are all synaptic potentials generated by the opening of channels through the binding of neurotransmitter. Whereas in the conductal region, we have specialized channels that are opened by a change in voltage, and the action potentials will propagate long distances along these axonal regions, along these conductor regions, to the output region, where the action potential will signal the opening of a voltage-gated calcium channel to release through the process of exocytosis of neurotransmitter. So any questions from there? So what we have so far is the upper panel, we have an anatomic description that could have been done, probably in 1900. And here we have a functional description that took another 60 years or so to sort out, because they had to sort out the electrophysiology of each of these different channel types. And then what I want you to hold on to from this, though, is that these are electrical signals. These are electrical signals that propagate at rates of about 1 to as much as 110 meters per second. Why I want to point this out now is that this electrical signaling, we're going to spend a lot of time on, this electrical signaling is confined to the membranes. Because where we're headed now is I want to start thinking about what's happening in the cytoplasm. And as we work our way through all of this and we think about neurons, you sort of have to keep your thinking separate. You have to think about what's going on at the membrane because that'll be electrical stuff going really fast. And then in the cytoplasm, we're going to have protein stuff going on, as we described earlier, fairly slowly. So now let's move into the cytoplasm and think about what's going on in the 99.9% of the cell, which is contained within the boundaries of the plasma membrane. So any questions to there? Well, let's take the anatomic description from above. We can move through this fairly quickly. And one of the obvious upshots of the anatomic description is that the neuron is a remarkably asymmetric cell. That if we were to put dimensions to some of these structures, we would find that typically a neuronal cell body would have a diameter of, this diameter of the cell body may be 30 to at most 50 micrometers in diameter. These dendrites could be about 1 to 2 millimeters. But the axons, in some neurons, the axons can reach greater than 1 meter. Now, one of the important concepts here is that all protein synthesis is in the neuronal cell body. And again, we find an important concept here of the different types of protein synthesis. Then when we talk about protein synthesis can be divided into cytoplasmic proteins, and can be divided also into vesicular proteins. Now, of course, vesicular proteins include those proteins that are packaged inside the lumen of the vesicle, as well as those that are across the plasma membrane, across the vesicular membrane. So the ones we've talked about so far were the ones we talked about where there was a Golgi apparatus and an ER. So we had the endoplasmic reticulum in our shorthand here. There were the ribosomes attached to the ER generating rough ER, and there were some ribosomes out here that were free ribosomes. But those ribosomes that were stuck on the endoplasmic reticulum, generating the rough endoplasmic reticulum, would produce proteins that were packaged into vesicles and then processed through the Golgi. And emerged as vesicles. But since the vesicles are simply cargo-carrying containers, you have to recognize that their genesis, their origin, is always up here in the cell body. That's where they start. And then all along the axon were these structures we talked about earlier. Called microtubules. And through a lot of anatomy and a lot of physiology and a lot of cell biology, it was found that the microtubules support kinesin and dynein-driven transport. Remember, these are the molecular motors. The kinesin is a plus end-directed motor, the dynein is a minus end-directed motor, so we can now imagine how these vesicles can be attached to the microtubules by kinesin and move in this direction. It turns out that the movement by kinesin and dynein, this movement, this so-called axoplasmic transport of vesicles and their proteins axoplasmic transport of vesicles and the proteins progresses in the neighborhood of about 200 to 400 millimeters per day. Now, I'll give you these numbers, but I really want to explain how they came to these conclusions. Proteins that are cytoplasmic proteins free in the ribosome, that are generated by free ribosomes, these proteins, let's spell it out over here, cytoplasmic proteins are transported at a rate of about 0.5 to at most 2 millimeters per day. So we now see there's a discrepancy, an enormous discrepancy in the rates at which cytoplasmic proteins and vesicular proteins are transported. One moves at about 200 to 400 millimeters a day, and the other moves at about 0.2 millimeters a day. In the last few minutes, let me describe the early experiments of how this was sorted out, because I think they're elegant experiments. What these scientists did is they recognized that they could dissect out of a rabbit retina. And the insight about the rabbit retina is that there were certain cells in the retina that would send processes from the retina into the structure here called the optic nerve. By understanding the anatomy, they recognized that these are the ganglion cell bodies. But that these cells had these great long axons that would extend into the brain, extend into a region of the brain called the hypothalamus. And that these neurons now had their output regions or axon terminals here in the hypothalamus. So now they had the three major parts of the neuron we described above. They had the neural cell body, the axon, and the axon terminal. They knew exactly where they were. The other advantage is out here in the retina, you have pretty good access to it. So what they would do is they would take a needle and they would thrust the needle into the eyeball and they would inject radioactive amino acids. These radioactive amino acids then are converted to proteins. And what I'll leave you with here before we come back to it tomorrow is that these proteins now are going to be transported from the neuronal cell body toward their output regions here in the hypothalamus. They could then wait certain periods of time, cut the optic nerve into pieces, and decide which proteins were moving at what rates. We'll take that up tomorrow, and that was the first initial description of axoplasmic transport rates.